No need to stand in long queue anymore for COVID test. Now you can do the test in the home. Sounds interesting? Then stay with me to know in details about it. Hello my dear friends, I am Poonam and you are watching Biology Explained. Coviself is India's first rapid antigen test kit approved by ICMR. Coviself can be used at home by yourself. How to test? Do I need to be a medical professional for testing? Is it very expensive? Everyone needs to do the test? How does it work? Wait. I will tell you everything in details today. So let's start with how does it work. Coviself is a rapid antigen test kit. That is, it will test the presence of antigen, and in this case, the antigen is SARS-CoV-2 virus, that is the virus causing COVID-19 disease. And this test will be done only in 15 minutes. One Coviself kit will contain a detailed instruction manual, a nasal swab, a buffer solution, a cassette, and a biohazard disposal bag. Let me walk you through the steps now. Download the Coviself app from your mobile. Fill all the important information. Step 2. Keep the test kit material in a well sanitized table. Step 3. Wash your hand and let it dry. Step 4. Take the buffer solution and tap it 10 times slowly so that the solution comes down completely before you start the test. Step 5. Open the nasal swab. The nasal swab has one stick side, which is the back side of the nasal swab, and a swollen side, which is the front side of the nasal swab. And you have to open the nasal swab from the tail or the stick side. The front side will have brush-like structure. Don't touch that side. Open from the tail or the stick side. Step 6. Insert the brush-like side inside your nostril 2 to 4 cm inside or until you feel some kind of resistance when you insert it. Remember, in this kit, you need to take the samples from the nostril and not from the nasopharyngeal region, which is done in case of RT-PCR. Now rotate the swab inside the nostril 5 times. Remember, the swab should touch the nostril end nicely. So you should be very careful doing this step. Take out the swab and insert it in the other nostril and do the same complete rotation 5 times. Step 7. Take the swab and insert it inside the buffer solution. Rotate the swab for 10 times and then break the swab stick carefully from the notch in the stick. Do it carefully so that the buffer does not spill outside. Close the buffer tube. Step 8. Now add 2 drops of this solution in the cassette. You have to add it in the round well of the cassette. You can see the cassette has a T and a C line along the horizontal side. Wait for 15 minutes and then you can see the result. Step 9. Take a photo of the cassette in your mobile through Coviself ad and then the app will tell you the results. Step 10. Now carefully put everything in the biohazard bag provided by the Coviself kit and dispose it carefully in a proper place. Please do not throw it anywhere. This can lead to contamination. Now let's interpret the result. 4 possibilities are there. You can get 2 lines on T and another on C. You can get 1 line only in C. You can get 1 line only in T, or you might not get any line. Let's understand each situation. When there are 2 lines, that is one on T and the other on C, it means firstly that the test had worked properly and that's the reason you see the C line. And since there is a line in the T, that means you are positive for COVID-19. So if 2 line comes, then 1 is positive for COVID-19. Next situation. As I have told you, if only C line is present, that is only control line is present, that means the test has occurred properly. But since there is no T line, that means there is no SARS-CoV-2 virus in the nasal swab, that is, you are negative for COVID-19. So, no T line, only C line means COVID negative. Now, situation 3 and 4 has no C line, that is, the control line is absent. If the control line is absent, then the test is invalid. You cannot accept the results even if you get a line in the T region. So, situation 3 and 4 says that the test is faulty. You need to repeat the test again with a new set of test kit. So, now you know how to do the test in the home. Anyone can do the test by oneself. You no need to be a medical professional to perform the test. So, it's doable by every common man. The price will be 250 rupees for each kit and it will be available from June in India. According to ICMR guidelines, everyone is not supposed to do the test. If you have symptoms of COVID-19, then you should perform the test. If you get a positive result, then you no need to reconfirm through RT-PCR again. You can consult your doctor and start the medication. But if you get negative results and still have symptoms of COVID-19, you have to reconfirm through RT-PCR. So now you know everything about Coviself. Let's talk a bit more about science here. If you want to leave the video, please do me a favor. Hit the like button and subscribe for more updates. For those who are staying with me, let's continue. First question is, why are we rotating the swab in the nose? So when we are rotating the nasal swab against our nasal cavity, we are actually collecting the viral sample. We all know SARS-CoV-2 virus enters through the nose. So if SARS-CoV-2 is present, it will get attached to the nasal swab. Next, we insert the swab inside the buffer. The buffer is nothing but lysis buffer. So it will lyse the virus into small particles. Next, we add the solution inside the cassette. Here very interesting things happen. Here lateral flow test is taking place. This cassette has a place where we add the sample and this is called sample pad. Next to it is the conjugate pad. Here antibody against the antigen, in this case antibody against SARS-CoV-2 virus is kept and these antibodies are called primary antibody. They have a color tag which will only be expressed when it binds to a particular antibody. These antibodies can move through the pad through capillary action. The technology which is used in this method is based on ELISA, but I am not going into the details of ELISA here. Next membrane is nitrocellulose membrane, through which the sample can move by capillary action. At last, we have the absorbent pad where the excess liquid is absorbed. Now, the nitrocellulose membrane has two types of antibody. One set of antibody is present on the T line. They are the same antibody as the antibodies present in the conjugate pad, but they are fixed, that is immobilized, and do not have any color tag. The second type of antibody is secondary antibody and they are present on the control line. This antibody will bind to the primary antibody of the conjugate pad only when they do not bind to the antigen. Let's consider a situation where the antigen, that is SARS-CoV-2 virus, is present in the buffer solution. So we are adding the solution in the well. The liquid will move due to capillary action. Now it will meet the SARS-CoV-2 antibody in the conjugate pad. So the antibody will bind to the antigen. However, there will be always some antibody which won't bind to the antigen since antibody is always in excess. Now all the bound and unbound antibody will move through the nitrocellulose membrane. Remember, the T line has specific antibody which can recognize SARS-CoV-2 antigen. So the antibodies moving with SARS-CoV-2 antigen will bind to it and color line will be produced at T line. The unbound antibody will move further and bind to secondary antibody on the C line and produce a colored line. The C place has antibodies which are not specific for SARS-CoV-2 antigen. So it will bind to any antibody and give the color line. So if the test work fine, antibodies will move and always bind to the secondary antibody. So we should always get C line, that is control line, and then only the test is considered valid. If C line is not there, it means that the antibodies are either not moving or there is some other reason why the color is not observed. So the test is faulty. So remember, we need at least C line to consider the test valid. Can you guess what will happen when the sample do not contain SARS-CoV-2 virus? Let's see. So again, we will add the sample in the well. Now the sample will move to the conjugate pad. But since no antigen is there, nothing will bind to the primary antibody in the conjugate pad. However, the unbound antibody will move forward due to capillary action, but it won't bind to the antibodies on the T line since they are specific for SARS-CoV-2 antigen. So the antibody will move further and it will bind to the C line, that is the control line will be produced. If we have only C line, then it means one is negative for SARS-CoV-2 virus, that is negative for COVID-19. And remember, no C line means invalid test. Hope it's clear to you now. Let me know in the comment if you have any question. And thanks for staying with me till the end. So please hit the like button and subscribe my channel for more COVID-19 updates. Thank you.